would like to welcome you to our winter spring webinar series for today. We have Dr. Andrew Wood here, Paediatric Oncologist at Starship and Senior Research Fellow at the University of Auckland. And he's kindly given up his time to come and talk to us about childhood leukemias. So we'll hand the time over to Andy Wood. And I'm your host, Tim Maiflini, Regional Manager for Support Services here in the Northern Region. All right, take it away, Andy.
Thanks for the intro, Tim. Welcome, everybody. So I was just going to chat about childhood leukemias. And, you know, the key question that struck me when I first saw children with cancer and when I see families with cancer is this question about, you know, why is this happening? Can this really be happening? And what I want to cover today is just a bit of stuff on pediatric cancer in New Zealand. How common is it? I want to talk about what is cancer, what is leukemia, and particularly the vexing question of why do young people get leukemia. I'll talk about the most common leukemia we see in pediatrics, which is ALL, or acute lymphoblastic leukemia. I'll talk about acute myeloid leukemia and some local research we've got happening at the uni here. So in the developed world, about 1 in 500 kids will get cancer before they turn age 15 years. And cancer is the most common disease cause of death from age 1 to about 40. Every week in New Zealand, we get about 3 new cases on average between Auckland and Christchurch. And of those 3, 1 of them is going to be a leukemia. The overall survival for all cancers in New Zealand is about 84%, 5 years after diagnosis. And obviously that's not good enough, but that does benchmark with our neighbours. And those survivals, you know, a third of those are made up from kids with leukemia. So one of the key questions is, what is cancer and leukemia? And cancer is actually intrinsic to our biology. People have found fossils with dinosaur bones, and people have found scrolls from the ancient world from lots of different cultures where people describe masses growing that were cancer. So that's been known for a long time. And normally in our body, we have all these cells, trillions of cells, and these cells are healthy, they have a function, they grow and they function it, they die, and they do it at the right time in the right place. That cancer is when you have an uncontrolled proliferation of abnormal cells. So it starts from a healthy cell, but the abnormal cancer cells then disrupt the healthy cells, and that causes dis-ease, and untreated, it will lead to death. Leukemia took a lot longer to discover than solid tumors. So big solid tumors that caused a big lump somewhere were pretty obvious, but leukemia was only really discovered once the microscope was discovered a couple hundred years ago. And what they noticed is that when you looked at blood under a microscope, you saw that it wasn't just red stuff, it was made of different cells. And if you spin the blood, you actually get you get all this red stuff, which is the red blood cells that carry oxygen, and you get a very, very faint line that doesn't project well on the screen here of white cells that constitute under 1% of all the cells in the blood and what we call the total mass of blood. When you get leukemia, you actually get a lot of the time an increase in the white blood cells. So here is a sample with the yellow arrow where the band of white cells, the leukemia cells, has increased. So here the band is tiny, barely visible, and here the white band is really obvious. Here, the healthy bone marrow is making lots of red blood cells, but in the patient with leukemia, the healthy bone marrow is not making many red blood cells, so there's a lot fewer red blood cells in the person that's got leukemia, and they'll feel tired and they'll look pale because of that. So the next question is, why does cancer occur? So the code of life is DNA in each of the cells of our body, and you get 3 billion letters from your mum and 3 billion letters from your dad. And every time a cell is copied, when you go from a parent to a child, or you go from a single cell embryo to an adult, all that DNA has to be copied. So you're copying 6 billion letters of DNA, trillions and trillions of times. And not surprisingly, you can get spelling mistakes happen. And spelling mistakes in DNA are called mutations. Now, the genetic origin of cancer being due to DNA mutations, people theorized about that a long time ago, but it was completely unproven. And then a woman called Janet Rowley applied knowledge about Down syndrome to cancer. And she discovered mutations occurred in cancer. And really that was the evidence that mutations could actually cause cancer. And she also showed that several mutations were needed to turn a healthy cell into a cancer cell. And here she is getting one of the presidential medals from Obama for all that wonderful work. So what she did is in children with Down syndrome and trisomy 21, they have 3 copies of chromosome 21. So these are all the 3 billion letters of DNA organized into 23 pairs of chromosomes. The last one's are gender chromosomes. Number 1 is the longest, 22 is the shortest. In trisomy 21, instead of 2 copies, you have 3 copies. And so Janet Rowley knew that you had 3 copies of these chromosomes in children with Down syndrome, or people with Down syndrome. Then she looked at a cell from a child with acute myeloid leukemia, and she said, hang on a sec, one of these chromosome 8s is a bit short, but one of these chromosome 21s is a bit long. So these chromosomes have been swapped around, and she found that in lots of AML, and she worked out that these bits of chromosome in the wrong location were called translocations, and that mucked up the code of life, and it was one of the steps leading to a cancer. So why do you get cancer mutations? Well, in adults, the big one is environmental exposures, which is UV light and smoking. You can inherit some cancer mutations. So one of the most famous is Angelina Jolie having a double mastectomy due to having the BRCA mutations. But those aren't big factors in kids. In kids' leukemia, the biggest factor is just plain old terrible luck. And so different types of mutations can be hereditary. You get them from your parents. They're replicative, which means bad luck when you copy cells, or they're from your environment. So lung cancer in the food pipe in the mouth is sort of from smoking causing cancer. But there's a few with faint red here where there's a low percentage of cancers are caused by genes you inherit from your parents. But the majority of cancers, including leukemias here signified as LK, the driving mutations that cause the cancer are bad luck. And in leukemia, that's approaching 100%. So leukemia mutations are really random bad luck. So in your blood, you have something called a stem cell, and that grows into different types of cells in your body with different types of jobs. So the stem cells make new cells, and every time you go to copy the DNA, there's a chance of a mutation. Now, in the average person, you acquire mutations pretty slowly. You'll acquire mutations in your stem cells, once mutation every year, but most of those mutations are actually harmless. Most of those mutations have no good function or no bad function. They just kind of sit there. Now, adults have more time to accumulate these mutations, and that's one of the key reasons why adults get more cancer than kids, that along with environmental exposures, which are often lifestyle choices. So why do young people get leukemia? So there's a guy from the UK called Mel Greaves, and his theory goes that you sometimes are born with the first mutation. So there's a really common mutation that's got a fancy name, EPP6 RUNX1, that's fairly common in B-cell acute lymphoblastic leukemia. But you also find it in 1% of healthy newborn babies, which is pretty crazy. But those babies don't have cancer. They just have cells in their body, some of which have a cancer mutation. Now, of those 1% of babies, only 1% of those will go on to develop B-cell leukemia driven by the same mutation, ETV6. That happens because their cells acquire extra mutations. So the babies are born with the first step, and over the first few years of their life, they'll acquire other mutations. But the vast majority of babies don't acquire those extra mutations, so they don't become cancerous. And other times you can have the cancerous mutations, but your body actually works out those cells are abnormal and it just destroys them. And sometimes we have cases where we've tracked those abnormal mutations, but a child gets a serious infection, and as the immune system's wiping out the infection, it also wipes out these pre-leukemic cells, which is a pretty crazy thought. So childhood leukemia you've got these diverse types of blood cells with different jobs. You've got red blood cells that carry oxygen around the body. You've got platelets that stop you bleeding. They form a brick wall in a blood vessel if you're bleeding. You've got these myeloid related immune cells that do different functions, neutrophils that eat bad guys. These cells would often fight different types of infections like parasites. Then you've got what they call lymphoid cells, which is called the main classes of B, T, and natural killer. But this is a grossly oversimplified diagram. But these diverse types of blood cells with different jobs, if they acquire mutations, they can turn into diverse types of different leukemias. Now, the big types of leukemias is actually the myeloid leukemias that come from the myeloid stem cell. And the other type is called the acute lymphoblastic leukemias that come from the lymphoid stem cell. So the most common leukemia in childhood is called acute lymphoblastic leukemia. And in the 1950s, survival for this was 0, but now the survival for this disease is over 92%. And the current trend is that every year there's a 0.5% to 1% improvement in survival. That may slow down as the last 8% may be tougher to solve than the earlier percentage. But the majority of kids that survive acute lymphoblastic leukemia have good quality of life that they consider very worthwhile. And the way this big increase has happened is a whole lot of paradigms in cancer care, when applied to this disease, or that were developed in this disease, they've all worked. So risk stratification, that's treating a more aggressive leukemia more aggressively, or a less risky leukemia less aggressively. Clinical trials comparing different recipes of chemo, bone marrow transplants, and something called targeted therapies and immune therapies I'll talk about more. All these different strategies have kind of worked in ALL to take this from a lethal disease to a disease where 9 out of 10 kids and teens are actually surviving long term. So remission is the phrase when we say there's no evidence of cancer. And the longer your remission, the more likely it is you're truly cancer free. And you can use, some people use the phrase cure, which means, you know, you're totally free of cancer and hopefully free of side effects of the cancer and its treatment, but that's another issue. Now, there's one type of this leukemia, B-ALL, that we subclassify as called favorable standard risk. And there's a long story about what that subtype is, but for that subtype of leukemia, life expectancy exceeds 97% at 10 years. There might even be close to 99% now. And if you look at these kids that are treated with this type of leukemia, and you compare them to their brothers and sisters, they tend to have a similar quality of life. They're just as happy, the same chance of going to uni or getting a job, getting married or having kids. And so that's a cancer that we do a pretty good job of treating. There are other cancers where unfortunately we don't do such a great job. But, you know, how did this happen? And back in the 1950s, these kids didn't survive. And this is a picture of one of my mentors who's now in her sort of mid to late 80s. And, you know, when I asked her what it was like doing this job as a pediatric oncologist when she started, she just went silent for a long time and just said, you know, then she looked up and she goes, they all died. All the children died. And, you know, why is it so different? And so the key reason it's different is the invention of chemotherapy. So this chap is called Sidney Farber, and he looked at cancer cells under a microscope, and he wondered whether giving them folate would fix the leukemia, but folate actually made the leukemias grow faster, which was obviously bad. So he tried a chemical called an antifolate. It pretended to be folate, but it blocked the cell using folate. It was kind of like a booby trap that jammed up the machinery of the cancer cells. And the antifolates he invented and others invented were precursors of the modern drug methotrexate. And so in the post-World War II period, Sidney Farber achieved temporary remissions in ALL. They weren't cures, but he gave this methotrexate drug, and for a while, the leukemia would be undetectable, but it would come back. Over time, people invented new drugs, and they combined drugs in different recipes, and they compared different recipes. And that invented what we call the cooperative clinical trial groups. The example of this would be the Children's Oncology Group, which is a North American one. And so what you do is you compare 2 different recipes for treating a leukemia, and you see which one is best, and then you go with that, and then you invent a new recipe, and you would compare the winner of that to the new recipe. And by doing that over decades and decades, that's led to the outcomes we see now. And so the clinical trials we offer in New Zealand for ALL are principally from the USA, but we also offer things from the UK and work with the Australians. And there's actually a really significant investment in research at Starship, just so we can actually run these clinical trials. And one of the key things is most pediatric ALL is actually treated with chemo drugs that were invented between the 1950s and 1970s. And we've just learned to use these drugs more elegantly and use the right dose at the right time in the right patient. We've also found some new drugs. So what we call sort of old-fashioned chemotherapy or cytotoxic chemotherapy, that chemotherapy is really about killing a fast-dividing cancer cell. And so those cytotoxic chemo damages the DNA of cancer cells, or it damages the machinery of cancer cells. So when a cancer cell is growing fast, the cytotoxic chemotherapy makes that cancer cell self-destruct. But cytotoxic chemotherapy also hurts a healthy cell that's dividing rapidly. One of the analogies for this is it's a bit like firing a shotgun at a bullseye. You'll hit the bullseye, but you're going to cause collateral damage. And that's the side effects that the patients suffer through. There are new types of therapies called targeted therapy that are more akin to a sniper's rifle, that are more precise, and they only hurt the cancer cell, and hopefully they don't hurt any of the healthy cells or hurt them much less. These often inhibit cancer mutations that aren't present in healthy cells, or they use the immune system to kill cancer. Often these have less side effects, but not every single time. The big class of drugs that helped fight this, the targeted therapy that helped fight leukemias, were invented in the late 90s, early 2000s. And one of them is called Gleevec or imatinib, and that's used to treat Philadelphia-like ALL. And at the time, people were like, oh my gosh, this is revolutionary. And people thought it might be a magic bullet and it would work for all different types of cancer. Unfortunately, this class of drug targeting mutations only works for a subset of leukemias. For most leukemias, it doesn't work. But when it does work, it can be a really fantastic, very helpful addition to therapy. The next thing that's been most exciting in the last 10 years has been using the immune system to cure therapy. And optimism about immune therapy has also really increased, particularly in the last, 6 to 8 years. And the reason this has really increased is a technology called CAR T cells. And I know there was a talk several weeks ago on this, but I just want to recap the key parts of CAR T for people that missed that talk. And so CAR T is a chimeric antigen receptor T cell. T cells is a type of immune cell that we all have, and T immune cells kill cells that are unhealthy or kill cells infected with viruses. What you do with CAR T cells is you take out T cells from a person with B-cell leukemia. So you have to take the T cells out of their own body. You take those T cells and then you genetically engineer them with a receptor. And the receptor can detect leukemia cells of a particular type, and then it makes the T cell kill the leukemia. So you grow these cells up and you give them back to the same patient. So that whole process takes a couple of months, and it's very complex and it's very expensive. And in selected cases of incurable B-cell leukemia, depending on which study group we're talking about, there's probably a 40 to 70% chance of something we call intention to treat survival for selected cases, which means for people with no other options, that's absolutely incredible. This is a picture of a young woman called Emily Whitehead, and I actually saw her being treated in Philadelphia when she was the first child in the world to receive this therapy. She's no longer 7 years cancer-free. I think it's now just 8 years and a bit. But the therapy that was used to treat her was FDA approved about 3 years ago. It's made by Novartis and it's called Chimera. Unfortunately, we can't deliver that therapy here. I think we technically could do it, but Novartis won't let us at the moment. So we've at the moment sent a limited number of people to Australia who are giving this therapy Chimera. So this therapy Chimera is no longer experimental, but it's an incredibly important therapy to access for selected people. The other major type of leukemia that happens in kids is called acute myeloid leukemia. So while ALL therapy is often considered a marvel of modern medicine for many patients because it's revolutionized the outcomes, AML is actually comparatively much more frustrating. So overall survival for AML is roughly around 2/3 of patients. But improvement in AML has been much slower. And a lot of the ideas and the strategies that worked in ALL, they just haven't delivered gains or big gains in AML. A lot of things just haven't worked. There's been a lot of hard work, but it hasn't really delivered the results we'd hoped for. And so, you know, the thing about AML therapy is, you know, just to give you an analogy, ALL therapy goes for several years, and there's an intensive part that lasts for half a year, but it's not as intensive as any of the therapy you receive in AML. So AML therapy is really intensive. One analogy is to say it's like 4 rounds in a boxing ring with heavyweight boxers. So the chemo for AML is much more intensive. It can be much more brutal, not for everybody, but for some people. But the chemotherapy is literally drugs from the 1960s and 70s, like these 2 famous boxers here. And because the chemotherapy is so brutal, it takes out the healthy immune cells that protect us from infection. And so the chance of kids with AML dying from infection is actually far too common. So AML is just a much tougher disease to treat. And generally speaking, it's tougher on the patients. One of the analogies for AML therapy is it really depends on 2 drugs that are quite old, delivered in slightly different ways. So for ALL, we might use between 11 to 14 drugs for the average patient, and they're used in quite complex combinations in quite precise ways. In AML, we're really using 2 drugs. I sort of think of it like an old-fashioned salad where you're really getting the same core ingredients just rearranged slightly differently. And there's been a lot of people trialing drugs for AML, new drugs, but they rarely show consistent or major improvement. People who are researchers in this area have, you know, spent 30 years trying to test new drugs for this disease, and none of them have been really big wins. So, you know, why aren't these things working in AML? Well, that's a really complex and controversial question and set of answers, and different people will give you different answers. But a couple of themes that are recurring is AMLs vary a lot from person to person. They are more different and diverse, arguably, than ALL, and that makes it really hard to design these sniper rifle targeted therapies for these small groups of people with ALL. They're just so diverse. The next problem is AML cells are really similar to healthy blood cells. It's really hard to find a difference that you can really exploit. So when you give a therapy to hurt a cancer cell that's AML, you're always hurting the AML cells a lot, and that causes lots of side effects. It's really hard to study new drugs in pediatric AML for lots of reasons, and there's a trial we hope to open next year, which New Zealand's going to be a part of, called PEDAL. But it's also really hard to build models of AML that you can study in the lab to find new insights and develop new treatments. Because what you have to do is play around with things in the lab, often for decades, and really get them working well, and then you can move it to testing it in adults and children, and hopefully it provides a benefit. But that's quite hard to do in AML compared to other diseases. So talking about the local research we have here at Auckland that happens in my lab, there's really 2 themes. where we're trying to make models of AML to help us understand and treat this better. And we're doing this in something called zebrafish in mice. So zebrafish are called zebrafish because they've got stripes on them. They're a small tropical fish that's a couple of centimeters long. This is a complex slide that's for another talk, but you know, this film, Ocean's 11, there was 11 thieves that had to cooperate to steal stuff. And so one thief wouldn't have been enough. You needed a whole bunch of thieves. And so one of the interesting questions in AML is, how do the different cancer mutations in AML, how do they cooperate to cause AML? So what we do is we introduce genetic engineering into these fish. These zebrafish become sick, and here's the fish with bleeding, the same way that a cancer patient can get bleeding. And this leukemia also glows. You can see in the figure below it, you can see that the fish has got some bright green, and that's signs of the back of the fin, that's some of the cancer. And with different combinations of these genetics in these fish, the fish have a different chance of catching leukemia and a different chance of surviving. So we're trying to make some models of these different types of AML in fish so we can unravel why different mutations are easier or harder to treat with our chemotherapy. The other thing we're doing is we're making a model of leukemia in mice. So there's a particular target on the surface of some AML cancers called mesothelin. And we've genetically engineered a mouse model, and we've made the cancer glow under certain conditions. And you can see on the left that the genetically engineered leukemia glows more over time when it progresses. But when we give standard chemotherapy, those same two drugs I talked about before, to the mice with the leukemia, the cancer almost goes away. The next thing we're going to do is test a drug that was invented in Seattle, especially for kids with leukemia. And it's a drug that searches out this protein called mesothelin, which children shouldn't have in their bodies at all. It finds it on the cell surface of AML cancer cells, and it delivers, and then it uses that to deliver a payload of chemotherapy just to the cancer cells and hopefully sparing the innocent cells. So we've made this model, and we're currently applying for funding to actually test this new drug that we're collaborating with Fred Hutch, which is a very famous cancer center in Seattle that invented bone marrow transplant, and they're pretty good at treating AML. The third thing that we want to introduce next year with treating AML is this trial called PEDAL, or Pediatric Acute Leukemias. And that's focused on AML, and that's an international trial across the US, Europe, and Australia, New Zealand. And the theory of this trial is that when we're trying recipe A, or first-line therapy, we're going to study a child's leukemia in greater depth and start working out what we could do for option B, C, and D ahead of time, just in case plan A doesn't work. It's a super, super complex trial with a lot of parts and a lot of different aspects to it. But what we're trying to do is study individual AMLs in more detail, and based on that, open clinical trials just in case a certain child needs them. So that's a very complex, expensive trial we're trying to lay the groundwork for now to try and treat it. And that antibody I was talking about before, we hope that new antibody treatment that we're going to test in mice, hopefully in New Zealand, is hopefully going to come through this pipeline in this trial here. So that's the end of my talk. Thanks everybody for listening. I'm not sure how the process works, but I'm very happy to take any questions.
All right, cool. So we have one question here that's on the Q&A section, and that question is, let's close your presentation. So, cool, all right. So first question was, how do families find out what the criteria is for having Chimera treatment in Australia?
So there are some criteria that people use, which is around that we don't think that standard leukemia therapy is going to be able to cure someone. So that's a situation where a stem cell transplant wouldn't be likely to work because it's such a complex therapy to manufacture, and the manufacturing, it takes a long time. You have to collect the cells, manufacture them, then inject them back. There's actually only a limited number of slots currently available in Australasia.
Mm-hmm.
But there's a group of people who treat leukemias, who have conferences across Australia and New Zealand, and anyone who in both countries who's going to be put forward for this therapy has to be presented to that committee, which is across all of Aussie and New Zealand, and all the committee has to agree that it's reasonable for that person to proceed, and they'll help make decisions about prioritizing people. So that's a discussion with someone's local primary hematologist or a hematologist that you see in clinic, and that's a question that you're welcome to ask. And then the other part of the parcel with this therapy is funding it. And the funding is an incredible challenge, and it's also an incredible challenge around the world. This therapy, the actual cost is sort of commercially sensitive, but the cost of the therapy is around about $500,000 US for one infusion. And then you've got the cost of the clinical care. And that makes it extremely expensive. And so in New Zealand, there's a process where we would have to apply to the Ministry of Health, and the Ministry of Health would make a decision. So not any of the pediatric oncology doctors, but a process in the Ministry of Health. And so in our part of the world, there's a complex process. And then there's different processes in different parts of the world to access this therapy.
All right. Thank you, Andy. Our next question is from an anonymous attendee. And they ask, what is Novartis's involvement regarding this being accessible in New Zealand?
So Novartis has the license to offer this product, and there's a very complex process to license a center to manufacture, to sort of do what we call the harvest, and then to sort of deliver the cells. And so in the States, there's only a limited number of pediatric cancer hospitals that can deliver this therapy. And there's a much smaller number in Europe. And so, I think there is so ultimately that they decide. And the initial, we've been in discussions about trying to get this product available in New Zealand for a long time. And, one step of that happening is Novartis entering into discussions with the ministry. And at this stage, my understanding is that the therapy we won't be able to deliver this therapy in New Zealand, even though personally I believe technically we could.
Thanks for answering that. Next question was, that was so informative and understandable. Do you think there are enough supports for parents? Since you are on the front line, do you have any suggestions or thoughts on how parents can be supported better or in a different way?
That's a really great question, and it's an incredibly challenging question. And so, the woman I showed you before is one of my mentors in the States. That's a question that people like Audrey have been grappling with for 50 years. And, if we had infinite resource, we would have bigger rooms on the ward. We would have more time for each family. We would have more supports in the community, including more financial supports, because, families having cancer is financially tricky. We'd have support so that siblings could get more education to compensate for the time that parents spent with other siblings. And so, and also that support starts at diagnosis, and ideally it should go on as long as the family needs it during treatment and once treatment's finished. So I think we may not get it right all the time, but our whole team does try very hard. And I think, I'm also really grateful to Tim and others at LBC, because I think, LBC and CCF also try very hard to provide another source of support. And I also know we have many families where, independent of the hospitals or independent of LBC, some families get a lot of support from their local communities.
Awesome. Thanks, Andy.
We're also happy to take suggestions. If people have ideas about what we can do better, we're also happy to take those.
All right, cool. Got 3 more questions. Next one was, why is the cost so high for this sort of treatment? And I'm assuming they're referring to the CAR T-cell therapy.
So that's an excellent question. So the first part of that answer is that when you make a drug, some of the old drugs that were made in the 60s and 70s, they're quite simple to make. They're quite, it doesn't take a very sophisticated biochemist to make those molecules. But when you make them according to good manufacturing practice, and you filter out the impurities, and you make sure you don't have any chemicals in there you don't want, the costs go up dramatically. And in the last few decades, even for drugs that were made a long time ago, pharmaceutical companies have been charging ever larger premiums for the medications they make. So for all the drugs we deliver, the costs are really going up. So before we even get to CAR T, even drugs that have come off patent, a lot of drug companies aren't dropping their costs. Like imatinib, the cost of that has gone up since it came off patent, just because drug companies want a return on investment for the capital. Now, the problem with CAR T therapy is that it's individualized. So you can make a molecule that's like methotrexate, and you can give the same molecule to everyone around the world with ALL therapy, and it's the same. But if we took a CAR T cell from one child with leukemia and gave it to a different child, that child's body would have a terrible, strong, life-threatening reaction against that cell, because it would see the other child's cell as an invader, because that cell would attack their body. And so you can't give CAR T made from one child to another child. People are trying to do that. It's called universal CAR T. But the evidence that that works is something we await enthusiastically, because that would hopefully make it much more accessible. So then you have a therapy that's really customized for the individual child. As a background, there was a therapy that people tried to customize for individual men with prostate cancer. So that's an incredibly common cancer. And they built, a factory worth billions to try and make this individualized therapy for prostate cancer. And it just became economically unviable. So it's incredibly hard to do, even with a cancer that's really common. ALL, fortunately, is a rare cancer. And so you've got a small number of kids, but you still have to build the big factory. So when Novartis, licensed this therapy, no one was sure what it would cost. And people knew that the manufacture was incredibly complex to do well and reliably. People spent so much energy making that happen, but it's hard to do. Now, that therapy, when Novartis were going to sell it, they came out with the early pricing, which is roughly $500,000 US thereabouts. And when they came out, a lot of clinicians and families cried out and said, "That is crazy expensive. How can you do that? That's unethical." And at the same time, a lot of investment bankers and people with a lot of shares in Novartis came out and said, "That's crazy. How can you do that? That's unethical. At that cost per patient, the company's going to lose money. The therapy won't be sustainable, and you won't be able to invest in other drugs." And so, there's a complex interaction between all those different factors. So it is an expensive drug. But to be honest, there's a lot of drugs coming through now where the cost per patient is going to be in the order of a couple of $100,000 per patient. And that's challenging around the world to deliver that. But it is going to be particularly challenging for the New Zealand health system.
That's an amazing answer, Andy. I've got 2 more questions. My next one was just wondering what advice would you give to a family who are apprehensive about participating in a clinical trial?
So another great question. So the first thing to understand about clinical trials is they are voluntary. You don't have to do it. And if you don't go on the clinical trial, people are still going to offer you what we believe is the very best clinical care. And turning down the clinical trial doesn't make us angry, doesn't make us cross. It's just we're putting an option in front of you, and you as the parent are in the best position to decide whether the pros and cons of those options are the right thing for your child and the right thing for you to live with. And so an intelligent, brave family could make a decision not to go for a clinical trial. And an intelligent, brave family could also make a decision to enthusiastically embrace a clinical trial. Generally speaking, we recommend clinical trials. And here's the general reasoning. Over time, the experimental arm or the new recipe of clinical trials has often been better than the old recipe. Now, it differs for different diseases, but often the new recipes, they have tweaks in them, sometimes subtle, sometimes less subtle, that are often advances. But you don't really know. If you're comparing two recipes, if you knew which was the right recipe, you wouldn't have to do a trial. So there are often subtle differences. But generally speaking, over time, the clinical trials have found the right answer, and that's why survival's improved from 0 to, say, for ALL, over 90%. One of the other factors for clinical trials is it means that the child's sort of care is to some degree being double-checked somewhere else. And so the child's diagnostics and aspects of the treatment and toxicity reporting are being reviewed by international teams. So for some trials that I'm a part of seeing, people email in with questions about this has happened, what do we do? And so you've got a group of people work out the best advice. So you just have more brains thinking about a situation than you otherwise might get, potentially. So I think those are probably the major two benefits. But I think the key thing is, trial and not trial both have pros and cons, and the consent process is ongoing, and you can ask questions later. The tricky thing is when you start therapy, often you don't want to wait too long to get going. And so you don't have all the time in the world to learn about something and make a decision, and which is particularly hard when you've got the stressful situation of your child being diagnosed.
All right. We've got an extra two questions that have just popped up. Next one was, what are the long-term side effects of children who have gone through chemo and survived?
Another great question. So that varies by disease, treatment, and person. So some chemotherapy, so for things that are easier to treat, might need gentler chemotherapy, and there's less likelihood of harm. And some things that are difficult to treat might be more difficult with side effects too in the short term and long term. So it really varies. And also some kids can cruise through a therapy where other kids have a really bumpy ride, and an individual child's presentation of cancer might have done them harm. So some cancers harm the child, and that harm doesn't necessarily disappear once you remove the cancer. And then, different children react to drugs differently. So it's really a tailored question on the individual. There are general guidelines. And to help with that process, we try and follow kids up in Auckland or Christchurch and in the regions. And we kind of look out for those things. And there's also information when kids are diagnosed about chemotherapy drugs and things to look out for long term. The other good news I would say is when we look at trials that were done 30 years ago compared to 10 years ago, we've taken away, where possible, therapies that cause a lot of long-term harm. And so the big things we worried about, like the initial disease coming back, damage to the heart and causing a different cancer, the risks of those things are actually dropping over time as a whole. But for each child, it can be different.
Next question was, are there any signals parents can look out for to catch any of these cancers early?
So that's tricky, and you know, a lot of us who treat these families, we're also got kids ourselves, and I think, you know, we'll all know that there's the odd thing that you sometimes wonder about or get anxious about. So the first thing is, child cancer is rare. The vast majority of kids who are tired or have a fever do not have cancer. The average GP will not diagnose, will never see a first presentation of child cancer in their life, in their whole career. And you think of all the kids coming to them per week. I think the key things with cancer presentations is that over time, the symptoms may wax and wane, they may go up and down, but over time they get worse. So if the child is experiencing something and its things just aren't quite right and it's not quite going away, or it doesn't go away completely, and it's getting worse over time, I think that's when you go to the GP. And different kids are different. In leukemias, is often fever, tiredness, looking paler compared to their brothers and sisters. But it's often, sometimes it can happen quite fast with a leukemia, like a child's well in the morning, and then by the afternoon, they're very sick. Other times it can be a couple of days or a couple of weeks. And once in a while, we see an acute leukemia where someone says, actually, I don't think they've been 100% for months. And so I guess the key thing is, if things aren't quite right, trust your intuition and go and get some advice, or go and get seeing GP. And if one GP doesn't quite reassure you, then think about going again, saying what your concerns are, or seeing another doctor who might have a different perspective on things.
All right. Thanks, Andy. There's one more question, and then I just have one question from me,
Yep.
if that's okay. Following on from the previous question about, I guess, late effects, what's the risk of secondary cancers for kids?
Yeah, so the secondary cancer is not the same cancer that the child initially had, but it's a different cancer. And that's actually really complex. So someone with the genetics or environment that leads to an initial cancer is probably at an increased risk of another cancer compared to the general population. That risk could be really small. That risk could be really tiny, but it may be there. But once in a while, in some families with the hereditary leukemias, that risk can be quite high of the second cancer anyway. But some of the cancer therapies we have, particular chemo drugs or radiation used for transplant, they can damage healthy cells' DNA, and that can lead to a second cancer. We try and minimize those therapies that cause second cancer as much as possible. So for AML, we tried to take out a drug called etoposide, for instance. The current clinical trial tries to avoid that. So we'll learn in a few years whether that was, you know, what the effects of that are. But it really does vary. In general, secondary cancers are a much lower risk to a patient than the original cancer. So treating the original cancer properly is still the right decision for the majority of cases.
All right. All right. So I guess I deal with lots of the parents that come up to Starship, and they're always worried when they first get their diagnosis and things. And one of the things that they kind of bring up is the fear of, hey, did I catch this early enough, or was there something that I could have done differently that might have been able to affect the prognosis of my child? And so does that have an effect how early you catch the cancer, and just trying to get a feel of whether parents are making the right decisions and those kind of things as well?
So for the vast majority with leukemia, I don't think time to diagnosis affects outcome.
Okay.
And the reason is that I think outcome mostly depends on the genetics of the leukemia itself, which related to the years and months over which the leukemia formed. And again, unless you're really, living around Chernobyl or Fukushima nuclear accidents, or, you worked in a toxic paint factory without protection in the 1960s, there's not things that parents are really doing that we understand that are actually causing leukemias. It's really those drastic radiation exposures that we know cause leukemias that are not really relevant for the majority. The biggest issue with treating cancer is, what are the genetics of the cancer and how does that respond to chemotherapy in the first month or 3 months? Those things are the things that really predict the likelihood of long-term survival. And those two things are not really related to time to diagnosis for leukemia. Because the leukemia is in the blood, and the blood goes around our whole body, the leukemia is kind of everywhere to start with, generally speaking. So hopefully that reassures people. And the thing about leukemias is sometimes they're really sneaky. Sometimes kids are still running around playing rugby, with leukemia. And I've seen these stories where kids look, where kids are slightly unwell, and then there's like 3 days where they suddenly get unwell really fast. And the parents kick themselves about, why didn't I notice? But, the vast majority of children who are a little bit unwell do not have leukemia. And so, as I said, a lot of us have our own, kids. And I would say that, the majority of us actually do not worry day to day with fever. Like if my kids get a fever, I don't think about leukemia, because leukemia is still so rare. It's really that recurrent process over time, progressive symptoms with kids getting worse and not being themselves. And to be honest, my experience of GPs is they get it right a remarkable amount of the time, given how rarely they see stuff. I know that's not always the case, but in general, I've been pretty impressed.
That's amazing. That's just a really awesome answer just for the parents that we support. The last thing that parents wanted to know was, and that's something that you probably come across all the time, and something that parents always talk to me and then talk to each other about, what things can they do to help their kids to be healthy. Like a lot of times they'll have family members that come and say, hey, you can take this kind of supplement and it will help you to do a little bit better.
Yeah.
What's your final thoughts on those kind of things?
So I think the key thing about looking after kids is for parents to trust their own instincts. I think the first step is when kids take their child home for the first time and they're super worried, well, I recognize that my child's sick. And in general, we find it remarkable how good parents are at spotting when their child is sick. And that often comes back to that intuition and sort of trusting your love and intuition for your own, for the kids you care for. I think understanding that education and familiarity with treatment does take time and you have a team to work with you. I think generally parents do a great job about trying to keep kids away from bugs and sickness. I know parents try really hard, and I think one of the reasons why COVID hasn't had such the impact we feared on kids with cancer is that families of kids with cancer were already super good at hand hygiene and super good at staying away from sick people. So they had been doing physical distancing for decades, well before COVID came along. And the next thing is about diets and supplements. And so there's actually some very recent data that just says a healthy, balanced diet actually leads to children feeling better and having fewer side effects during treatment, but it doesn't decrease the effectiveness of cancer therapy. So if, when your kid has cancer, getting them to eat healthy food and veggies isn't always that easy, with nausea and vomiting, but it's worth trying to find something if you can. The supplements we give are pretty balanced. We try to bring them pretty good, but a healthy, balanced diet is actually the best thing. One thing is, people often ask about things like really high dose vitamin supplements, and particularly vitamin C is the one that keeps on coming around. People talk about vitamin D and these things, and there's a long list of different chemicals and supplements, some in general use, some particular to different types of leukemias. I don't really think there's adequate evidence that those things are home run helpful yet. Something like, say, vitamin C, there's also a chance that high dose vitamin C could actually stop a lot of chemotherapy actually working that well too. And so the guy that discovered the shape of DNA, you know, he's a very famous researcher for obvious reasons. And he also did work showing that sometimes those high dose supplements could make some cancers progress faster. You know, some people are trying things like high dose vitamin C in very particular clinical settings with very particular types of cancer, often in combination with very particular types of chemotherapy. So an Aml therapy with very, very high dose vitamin C continuous infusions. But no one knows if that works yet. And that stuff often led by places like NYU, that's very, very early. And I think it's a long, long way from being a slam dunk. I think the vast majority of the evidence and experience is that a really good, healthy, balanced diet. If parents want to do a little bit more and they can afford it, I sometimes say, look, maybe go for organics, but there's no evidence it helps. But if you fully want to do extra, but if you can't afford organic food because it's so expensive, I don't think there's any reason to feel guilty whatsoever. So, you know, coming back to it, asking questions, getting educated, trusting your intuition, ringing to ask questions, speaking to doctors, friends, families, guys like yourselves, a healthy, balanced diet. And then I think the other key thing I'd say for parents is to look after yourselves. Looking after a child with cancer is beyond a full-time job when you already often have full-time jobs. And I think the other best thing you can do to support your kid is to look after yourself and not to feel guilty about it. And, you know, circle the wagons, bring in your friends and family, take a break. If someone offers you a few hours off, take a few hours off, go for a walk with your partner or your friends, go to a movie, as best you can organize. I think that's also key because it's a marathon. So I think that's my general advice.
Andy, that's definitely amazing advice, and I appreciate not just me, but leukemia and blood cancer and all of our patients and families really appreciate your time and the expertise that you've shared today, as well as the advice you've shared for our parents, because our parents are the ones that kind of hold these kids and look after them and kind of bear all the brunt in terms of stress and anxiety.
Absolutely, absolutely.
And I know that they're very, very grateful at this time. We're super grateful for you coming along to present today. Yeah, I wish you the best for the rest of your day and whatever else you're doing today. And look forward to catching up with you again.
Cheers, Tim. Thank you for everything you guys do and to all the parents for joining in. Thanks, everybody.
All right. Cheers. See you later.
Bye-bye.